Dear students, good morning. This is Dr. B. Dhanalakshmi, Assistant Professor, Department of Zoology, Nirmala College for Women. Here to discuss you about the topic enzyme-linked immunosorbent assay, methods and its application. ELISA. ELISA means enzyme-linked immunosorbent assay. It is an antigen-antibody reaction. In 1971, ELISA was introduced by Peter Perlman and Eva Engvall at Stockholm University in Sweden. It is a common laboratory technique which is usually used to measure the concentration of antibodies or antigens in blood in case of certain infectious disease. ELISA is the basic assay technique known as enzyme-linked immunosorbent assay. This is a quantitative analytical method that shows antigen-antibody reactions through the color change obtained by using an enzyme-linked conjugate and enzyme substrate, which serves to identify the presence and concentration of molecules in biological fluid. This plate-based assay technique is designed for detecting and quantifying substances of very low concentration molecules, such as peptides, proteins, hormones, vitamins, and drugs. In this assay, an enzyme conjugated with an antibody reacts with colorless substrate to generate a colored product. Such substrate is called chromogenic substance. So far, we have seen the introduction about ELISA. Now, we are going to look into the principle on which ELISA works. ELISA works on the principle that specific antibodies binds the target antigen. ELISA uses an enzyme to detect the binding of antigen antibody. It also helps to detect the presence and quantity of antigen binding. The enzyme converts a colorless substrate, which is called as chromogen, to a colored product, indicating the presence of antigen antibody binding and reaction. In order to increase the sensitivity and precision of the assay, the plate must be coated with antibodies with high affinity. Based on the principle, there are types of ELISA. ELISA test can be classified into three types, depending upon the different methods used for binding between antigen and antibody, namely direct ELISA, where antigen immobilized directly on the plate; indirect ELISA, antigen is coated to the microtiter well; sandwich ELISA, it is antibody is coated on the microtiter well; competitive ELISA, where microtiter well, which is antigen coated, is filled with the antigen antibody mixture. Now, we'll be seeing about direct ELISA. Here, the antigen is coated onto the well by passive adsorption, and later the antibody conjugated with the enzyme is added and incubated with the antigen, and after a period of incubation, the substrate or the chromatograph is ordered and the color develops. So this implies how the antigen is reacted with the antibody. This is the direct ELISA technique. The second one is indirect ELISA, where the antibody can be detected or quantitatively determined. Serum of some sample containing primary antibody is added to an antigen coated microtiter well and allowed to react with the antigen attached to the well. After any free antibody is washed, the presence of antibody bound to the antigen is detected by adding an enzyme conjugated secondary anti-isotope antibody, which binds to the primary antibody. Any free antibodies that is washed away and the substrate for the enzyme is added. The amount of colored reaction product that forms is measured by a specialized spectrophotometric plate reader, which can be measured and absorptions of all the wells of a 96 well plate in seconds. Indirect ELISA detects the presence of serum antibodies against human immunodeficiency virus. The third type is sandwich ELISA. Antigen can be detected by measured in this technique, the antibody is immobilized on a microtiter well. A sample containing antigen is added and allowed to react with the immobilized antibody. After the well is washed, a second enzyme-linked antibody specific to a different epitope on the antigen is added and allowed to react with the bound antigen. After any free second antibody is removed by washing, substrate is added and the colored reaction product is measured. The number of enzymes have been employed for ELISA includes alkaline phosphatases, horseradish peroxidases, galactosidases. These assays approach the sensitivity of radioimmunoassay and have the advantage of being safer and less costly. Now we look into the ELISA procedure. It is one of the easiest blood tests that can be carried out. It is a rapid, quick, and requires a blood sample of the patient. The entire procedure of ELISA is as follows. An antibody is attached to the polystyrene plate, which is a solid surface and is attracted or has an affinity towards bacteria, other antibodies, and hormones. A microtiter coated with the antigen is filled with this antigen-antibody mixture, after which free antibodies are removed by washing. A second antibody specific to primary antibody is added, which is usually conjugated with an enzyme. Free enzyme-linked secondary antibodies are removed by washing the plate. Finally, the substrate is added. The substrate is converted by the enzyme to form a colored product, which can be measured by spectrophotometry. HCG protein, which indicates pregnancy, is detected by ELISA. A combination of blood or urine sample and purified HCG linked to an enzyme is added to the system. If HCG is absent in the test sample, then only the linked enzyme binds to the solid surface. The more of the substance of interest is present, the more reaction takes place and less of linked enzyme binds to the solid surface. The reactions are indicated usually with a change in the color of the solution. Now we are going to see about the diseases that can be diagnosed using ELISA. ELISA can be used to detect some of these conditions: Ebola, pernicious anemia, AIDS, rotavirus, Lyme disease, syphilis, toxoplasmosis, Zika virus, carcinoma of the epithelial cells. Advantages of ELISAs are: it is easy to identify a particular kind of a disease. It is a rapid test which yields results quickly. It can be used to find out complex samples as the antigen is not required to get purified to detect. It is a highly responsive since direct and indirect analysis methods can be carried out. Possible detection for ELISA ranges from the quantitative, semi-quantitative, standard curves, qualitative, calibration curve models. Easier to perform and uncomplicated process as compared to other assays, which requires the presence of radioactive material. Advantages of ELISAs are: it is easy to identify a particular kind of a disease. It is a rapid test which yields results quickly. It can be used to find out complex samples as the antigen is not required to get purified to detect. It is a highly responsive since direct and indirect analysis methods can be carried out. Possible detection for ELISA ranges from the quantitative, semi-quantitative, standard curves, qualitative, calibration curve models. Easier to perform and uncomplicated process as compared to other assays, which requires the presence of radioactive material. Now we will be looking into the application of ELISA. The presence of antibodies and antigens in a sample can be determined. It is used in the food industry to detect any food allergens present, to determine the concentration of serum antibody in a virus test, during a disease outbreak to evaluate the spread of the disease. Example, during the recent COVID-19 outbreak, rapid testing kits are being used to determine the presence of antibodies in the blood samples. Thank you.